In the mid-6th century, the world was struck by one of history's deadliest pandemics, now known as the Justinian Plague. Named after the Byzantine Emperor Justinian I, who ruled from 527 to 565, this outbreak devastated cities across the Mediterranean and beyond, with death toll estimates ranging from 25 to 50 million people, nearly half of Europe's population at the time. Despite its historical significance, the exact nature, origins, and pathways of this plague have remained a mystery for centuries, leaving historians and epidemiologists to piece together clues from sparse historical records, mass graves, and anecdotal chronicles. However, the advent of ancient DNA analysis has revolutionized our understanding of this catastrophic event. By extracting and sequencing DNA from the teeth and bones of victims buried in mass graves, scientists have been able to identify the pathogen responsible, Yersinia pestis, the same bacterium behind the more infamous Black Death of the 14th century. This breakthrough has not only confirmed long-standing hypotheses about the cause of the Justinian Plague, but also allowed researchers to reconstruct its genetic makeup, revealing how it differs from later strains and how it may have evolved over time. Recent studies suggest that the strain responsible for the Justinian outbreak had unique genetic markers that made it particularly virulent and capable of rapid spread, shedding light on why the mortality rate was so devastatingly high. Moreover, analysis of ancient DNA provides insights into the transmission pathways of the disease, suggesting that it likely spread via both flea-infested rats and human-to-human contact, facilitated by the dense urban centers and active trade routes of the Byzantine Empire. These findings help explain the sudden, explosive nature of the epidemic and why it repeatedly resurged in waves over the next 2 centuries. Beyond identifying the pathogen, ancient DNA research also allows scientists to study the human immune response to the plague. By examining genetic material from victims, researchers can observe how human populations were affected, which genes may have offered resistance, and how these selective pressures may have shaped subsequent generations. This intersection of archaeology, genetics, and history creates a more nuanced understanding of the social and biological impact of the Justinian Plague. Historians have long debated the broader consequences of the pandemic, including its role in weakening the Byzantine Empire, altering trade networks, and reshaping demographic patterns across Europe, the Middle East, and North Africa. Ancient DNA evidence now provides a solid scientific foundation to contextualize these historical effects, linking the genetic evolution of the plague to observable patterns in population decline and recovery. Interestingly, comparisons with the Black Death strain reveal both similarities and differences in virulence and spread, offering a rare glimpse into how pandemics can evolve over time and how pathogens adapt to human hosts. This knowledge has modern implications as well, illustrating the importance of monitoring pathogen evolution and providing a historical lens for understanding contemporary infectious disease outbreaks. Experts caution that while ancient DNA offers unprecedented clarity, it is only one piece of the puzzle, and integrating historical, archaeological, and environmental data remains essential for a complete picture. Nevertheless, the unlocking of the Justinian Plague's secrets through ancient DNA represents a remarkable achievement, bridging centuries of human history with cutting-edge genetic science. It reminds us that the past, even events over a millennium old, can inform present understanding and guide future responses to epidemics. And that's something worth remembering when you next consider the resilience and vulnerability of human societies in the face of infection.